Thank you for joining this ESID Juniors Journal Club. I'm Laura Koval, and I'm a PhD student in the diagnostics of immunodeficiency. And more pertinently, perhaps, I'm the country representative for Sweden to the ESID Juniors as well. Today, it's my great pleasure to welcome as speaker my friend and colleague, Dr. Tessa Campbell. Tessa started her research career doing her PhD at the University of Sydney in Australia on interactions of NK cells and herpes viruses. In 2019, she then came to the Karolinska Institutet in Sweden to begin a postdoc on NK cell differentiation, NK cell deficiencies, and also more recently working with PDC diagnostic assays as well. Nonetheless, I assume her proudest accomplishment is being winner of the ESID 2022 Top Influencer Award. And today she's going to talk about her work characterizing IRF7 deficiency in patients who have had COVID-19 or other viral triggers. So I'm very excited. Go ahead.
Great. Thanks so much, Laura. That's the nicest introduction I think I've ever had. So that's a good way to start. Okay. Yes, so as Laura said, I'll be presenting today a recent publication from our group that was in collaboration with a lot of people looking at IRF7 deficiency in humans. And a bit different to normal, I actually wanted to start with the acknowledgements just to kind of put in context all the people that were involved in this study. So our group at the Karolinska Institutet was strongly supported by collaborators at the Karolinska University Hospital, and this was led by Yenan Bryceson and Peter Bergman. And then there was a team of us also involved in doing this work, and I particularly like to point out Laura, who was doing a lot of the genetic side of this project while I was tackling more of the immunology. So it was really a team effort here along with our clinical collaborators. And then this work was done in collaboration with Jean-Laurent Casanova's team in New York and Paris, and along with Isabel Mates's team in Belgium. And this work wouldn't have been possible without the COVID human genetic effort that really teamed our different labs together, combining our IRF7 deficient patients. And of course, we'd like to give our thanks to the patients and their families. We do this work with our patients for our patients, so it wouldn't be possible without them. And I'd also like to give thanks to the Swedish funding sources that we had that were able to make this work possible. So our paper really centers on the type 1 interferon response, and this is a really broad classical mark of antiviral immunity. So generally type 1 interferon is secreted by cells when they've become infected with a virus, and this leads to an antiviral state in bystander cells. So it triggers antiviral protective mechanisms in surrounding cells, which will stop them from getting or limit the amount of infection they'll contract, and thereby slowing down a viral infection. So it's a really important first line of defense or early defense against viral infection. And one of the main immune cells that's involved in also promoting the type 1 interferon response are a particular subset of dendritic cells called plasmacytoid dendritic cells. And these are quite an unusual dendritic cell type in that they have a lymphoid origin compared to myeloid progenitors that give rise to the majority of dendritic cells. And this really makes them quite unique in that they have a really high capacity to produce a huge amount of type 1 and also type 3 interferons upon stimulation. And despite being a very small population in peripheral blood, PDCs make up less than 1% of the white blood cells in the blood. They will produce the majority of type 1 interferon during a context of stimulation. So they're small, but they're very powerful. And in being able to produce type 1 interferon, PDCs, as well as other cells, are able to achieve production of type 1 and type 3 interferon through the transcription factor IRF7. And this leads to expression of both interferon alpha and interferon lambda, and also has partial involvement in interferon beta production. So our work really drew from some of the original case studies that were on IRF7 deficiency. So the first report of IRF7 deficiency was in 2015 of a young French girl. And with the onset of the COVID-19 pandemic, 2 more additional IRF7 deficient patients were identified and published by Zhang and colleagues from Casanova's group. And following on from that, we were able to identify 4 more additional patients. In particular, we focused on 2 brothers that came from Sweden and a young girl that came from Belgium. So in total, in our paper, we were focusing on collectively comparing all 7 patients, but particularly focusing on the 2 Swedish brothers and the Belgian girl, as these ones hadn't been immunologically or clinically characterized before, or genetically either. So in total, from the 7 IRF7 deficient patients, they came from 6 different families and 5 ancestries. And of the IRF7 variants that these patients carried, 5 were in a homozygous state and 2 were compound heterozygous. So we first wanted to validate the new IRF7 variants to see what the effect on IRF7 expression and function would be. And so we used an interferon beta luciferase reporter cell line, which was challenged with Sendai virus, which would drive interferon expression. So we can see in wild type cells here that when they're stimulated with Sendai virus, you get induction of interferon beta, which is detected by the luciferase reporter measured here. And in comparison to the index patient that had been previously described, we could see that this patient's 2 variants, when expressed, are complete loss of function. And when we contrasted this to the 3 new patients that we'd identified, patient 4, when their 2 variants were expressed together, this led to a loss of function, while one of them, when expressed alone, was still leading to interferon beta production, but in a compound heterozygous state, there was no interferon beta being produced. We could also see with patient 7 that her variant that was expressed homozygously led to no interferon beta, and the 2 Swedish brothers, their variant led to reduced but still some level of expression of interferon beta. So we then wanted to look in primary cells from the patient, and to do this, we took PBMCs that were stimulated with interferon beta for 24 hours, and this would lead to IRF7 expression. So you can see in the diagram on the left that IRF7 is kind of in a positive feedback loop with type 1 interferon. So initial levels of type 1 interferon will drive IRF7 upregulation, which leads to more type 1 interferon being produced, which stimulates more IRF7 and more type 1 interferon. So we can model the system by just stimulating cells up front with exogenous interferon, which will lead to upregulation of IRF7. So we can see in our 2 healthy samples, when we added interferon beta for 24 hours, this led to a really strong induction of IRF7, while in total PBMCs, there's undetectable levels when they're not stimulated. However, when we did the same treatment with PBMCs from patient 6, one of the Swedish brothers, we could see that there was no induction of IRF7, and the same was also true with patient 7 when stimulated with interferon beta. So despite the fact that there may be some residual function of IRF7 in the variant carried by the 2 Swedish brothers, in context, the IRF7 isn't being expressed at all by these cells, so it's undetectable. So we were able to conclude from this that the 4 new patients that we'd identified, that their variants of IRF7 all led to complete loss of function. So we then wanted to see what effect that this would have on the immune system. And so we focused in on these plasmacytoid dendritic cells as they basally express really high levels of IRF7. So you can see at the top on the left that if we compare IRF7 levels in T cells, NK cells, monocytes, and PDCs at the top, that there's somewhat intermediate level of expression of IRF7 in monocytes, very little in NK cells and T cells, but PDCs have really high levels of IRF7. And this is what you would see in healthy PBMCs. So we wanted to see what the outcome of loss of IRF7 expression would be in our patients. And to do this, we use stimulation with TLR7 and TLR9 agonists. So these two toll-like receptors are triggered during viral infection and other contexts where they can lead to signaling that includes IRF7, which leads to downstream cytokine expression. So first we looked at TLR7 stimulation, and to do this we used imiquimod, which is a TLR7 agonist. And we can see in the top row of the FACS plots that when we stimulate with TLR7, we get really strong induction of both interferon alpha and TNF expression. And you can see this quantified to the right, where looking at a range of healthy donors, we see a nice spectrum of interferon alpha being produced and a really high level of TNF being produced from healthy samples. However, when we looked at PDCs from one of the Swedish brothers, patient 5, we could see that there was pretty much no interferon alpha being produced at all, while there was still maintained expression of TNF. And again, this is quantified to the left, and we could also see the same for the other Swedish brother, and we looked at both brothers at 2 different time points, which makes up the 2 different points on the graph. So overall, we could see that with TLR7 stimulation, we got no interferon alpha production from PDCs in the patients, while we still had maintained TNF production. And this fits with what's known about the role of IRF7 in that it drives interferon alpha expression, but it's not involved in TNF, which is mainly driven through NF-kappa B downstream of TLR7. So this is really teasing apart the signaling pathways that move from TLR7 stimulation to cytokine responses. We also were able to look at samples from patient 7, which was the young Belgian girl. These samples had to be done on cryopreserved cells, which gives a much more reduced response for PDCs. They're a very sensitive cell type and quite difficult to work with. But we were still able to get a clear response from both the healthy controls in terms of looking at interferon alpha and TNF, although the rate of response is reduced compared to using fresh blood. However, we could see compared to frozen healthy controls that patient 7 had the same phenotype as the patients 5 and 6 from Sweden, in that she had basically no production of interferon alpha, while she had normal levels of TNF production. We then compared this to TLR9 stimulation. So this was using CpG-ODN, which is a TLR9 agonist. And similarly to TLR7, we could see that this stimulation leads to high levels of interferon alpha and also TNF in healthy controls, but we see no interferon alpha production in the patients, with either fresh samples from patients 5 and patient 6 or frozen samples from patient 7, while TNF production is mostly unchanged, as we could see in the fresh samples, although it was somewhat lower in patient 7 when we're looking at frozen cells. So overall from this, we could really see that loss of IRF7 in the patient's immune system meant that their PDCs were really depleted in their capacity to produce type 1 interferon. However, their ability to produce other cytokines such as TNF appears to be mostly unaffected. We're also able to confirm this more broadly by using single-cell RNA-seq of isolated peripheral blood PDCs that were challenged with either SARS-CoV-2 or influenza A virus. So this is just comparing patient 2 compared to a healthy control. You can focusing in on the cytokines that have been upregulated. So they're color marked by type 1 and type 3 interferons in red and yellow, ISG-related genes in purple, and inflammatory cytokines and markers in green. And you can really see that with both SARS-CoV-2 and influenza, the patients fail to upregulate these cytokines compared to the healthy controls. But I also wanted to draw your attention to interferon beta, which we can see here, which is still at a somewhat comparable level, or at least not as strongly reduced in the patient compared to the healthy control. And this has already been established that interferon beta is able to be expressed in the absence of IRF7 at a reduced but still detectable level. So then thinking about how this translates into the clinical features of our patients, it's really interesting to note that the majority of our patients had lived predominantly healthy lives, minus having one severe episode of pulmonary viral disease. So for the majority of these patients, the main virus that they suffered from was SARS-CoV-2. The second most common viral susceptibility was influenza. And then one patient had issues with adenovirus and respiratory syncytial virus. So mostly these patients are really able to tolerate the majority of infections and vaccinations that they've encountered, which is in contrast to other inborn errors of immunity in type 1 interferon, where there's a much broader spectrum of viruses and also vaccine-disseminated diseases that these patients are susceptible to. And this is probably because the other inborn errors of immunity in type 1 interferon affect more broadly the interferon response, while in the case of IRF7 deficiency, is really targeting type 1 and type 3 interferon specifically, while interferon beta remains. And we were also able to more broadly confirm that our patients had tolerated other viral infections by using VIR-scan, which screens for plasma levels of antibodies against a huge range of proteins that are expressed from known pathogens that may be encountered over the course of life. And we could see, compared to healthy controls, that our patients had comparable responses, indicating that they come into contact with many normal pathogens that you would expect. And this was also supported by conventional serology analysis. Oh, yeah, these were the patients highlighted at the end. So lastly, we wanted to see what the broader effect of having an IRF7 deficiency might have on the immune system, particularly as our lab is quite interested in the how the immune system is affected in cases of inborn errors of immunity. So we wanted to see if there would be an effect on T cells with the lack of IRF7 in these patients. So to do this, we use two different assays that I'll just walk you through. So the first one is a blast assay that was performed in the clinic here at Karolinska. And this uses whole virus from SARS-CoV-2 and the influenza split virion vaccine to then culture these stimuli with whole blood from patients and then look at the number of blasts that are formed. So basically enlarged T cells indicating that they've recognized the stimuli and have been activated. And we can see, compared to a healthy reference range of the number of blasts per microliter of whole blood that you would expect for CD4 T cells against influenza, that the patients had a significantly higher number of blasts being formed in response to influenza, although they had a normal number of blasts being formed in comparison to SARS-CoV-2 stimulation. And the healthy control reference range done here was with people that were known to have had SARS-CoV-2 but had had a mild infection. So we were quite interested to see that the patients had a much stronger response to influenza stimulation. And so to look at this more closely, we developed a peptide-specific memory T cell assay. And this works by taking T cells, stimulating them with specific peptides from different viruses, and then looking to see how many T cells from the memory compartment respond to that stimulation. So this gives an indication of how much of the T cell memory compartment is made up of T cells that are specific to that particular peptide. So we first benchmarked a number of healthy controls to see what would be a normal frequency of T cells that you would expect to be responding. And then we looked at our patient. And the readout here was interferon gamma responsiveness after stimulation with the peptide for several hours. And first, as our control, we had a stimulation with PMA ionomycin, which leads to a strong expression of interferon gamma, which we could see was comparable between the healthy controls in grey and the two Swedish patients in the black triangles. And then we compared to peptides that came from viruses that we knew the patients had tolerated quite easily. So we had several from human cytomegalovirus, or HCMV, and a combination peptide pool from Epstein-Barr virus, or EBV. And the patients here had similar levels of memory CD4 T cells that responded to these peptides, comparable to the healthy controls. However, when we looked at the response to influenza peptides, we could actually see that for several of these, the patients had a higher frequency of memory T cells that responded to influenza peptides. So particularly MP1, NA, and NP. The patients had a higher frequency of T cells that were able to detect these peptides compared to the healthy control. And the same was also true for the nucleocapsid protein from SARS-CoV-2, not so much for the spike protein, but for nucleocapsid, we could see this enhanced response. So this suggests that for the IRF7 patients, they have a larger frequency of CD4 T cells that are specific to influenza. And this is quite interesting because it may be a response from an interferon alpha feedback loop. So normally interferon alpha, as well as inducing an antiviral state, is involved in limiting immune pathology, so activating cells that will lead to their death as well, so that you don't have too much immune response, which can also lead to pathology during infection. But in the case of these IRF7 deficient patients, where they're not able to produce interferon alpha as efficiently, then they may be left with a larger pool of influenza-specific T cells or COVID-specific T cells after infection, and these might also be able to provide compensatory immunity so that if they came into contact with these viruses again, they would stand a better chance of tolerating them. We also looked at the CD8 T cells, oh, that's highlighted there, to compare back to the CD4 T cells, and here we saw a similar phenotype, but it wasn't as strong as we saw with the CD4 T cells. So one of the brothers had a very high blast response to influenza in the CD8 T cell compartment, but the other brother, it was not as high. And they both had within the healthy range for the SARS-CoV-2 response in terms of blast formation, which was the same as with the CD4 T cells. When we looked at the peptide-specific responses, we could see an increased frequency of memory CD8 T cells to peptides from both the nucleocapsid and the spike protein of SARS-CoV-2, again indicating that in the CD8 compartment, there's a higher frequency of memory T cells to COVID in these two brothers, which is quite interesting. So overall, in our paper, what we were able to show was that we were characterizing the IRF7 deficiency in 7 patients, 4 of which were previously undescribed, from 6 different kindreds and 5 different countries. So it was really a global effort to bring these patients together. And from our work, we showed that IRF7 deficiency was leading to diminished production of type 1 and type 3 interferon, but there still is residual interferon beta, as it being previously reported. And we think that this, along with the enhanced T cell immunity to viruses that these patients were particularly challenged with, leads to compensatory protection, explaining why these patients have mainly only had one really severe viral infection in their life, and it's particularly from viruses that are quite virulent, such as SARS-CoV-2 and influenza.